I'm Arash Rashed, a postdoc at Texas AgriLife Research in Amarillo. What I presented in Zebra Chip meeting this year was about psyllid movement, fluctuation in psyllid populations, how psyllid population change in natural vegetation in relation to potato emergence time, and what are the potential alternative hosts in the environment, and whether those alternative hosts for psyllids can act as a source for the pathogen or as a reservoir for the pathogen as well. We looked at these yellow sticky traps across Texas. We collected traps every 2 weeks, tested those psyllids for LSO presence, whether they're positive or negative. What we found, one common pattern across all these three multiple locations that we looked at was that psyllids first appear in natural vegetation. After that, we had three distinct patterns. First, we saw it in Pearsall location. Psyllid populations, they go up in natural vegetation, and then they drop down, and then there is a peak in potato field, and then again the population disappears after harvest. So here we had a peak in natural vegetation, but the biggest peak, and then it went down in natural vegetation. The biggest peak was in potato fields. The other pattern that we observed was in alternate spring lake location. We had the peak in natural vegetation, but it was the biggest peak. So an increase, a spike in psyllid population, a drop, and then we had a smaller peak in the potato fields, and they started moving, as potato emerged, they started moving into the potato fields. The final pattern that we saw was that potato psyllids in natural vegetation and in potato fields, they increased at the same time. But as I said earlier, in all these locations, always psyllids appeared in natural vegetation first. We looked at plants, the natural plants, potential natural hosts, tested them for LSO presence. They never tested positive so far. So this is a 2-year project. We did it for a few months. This is the first year, almost first year. We still have a couple of months to go. So far, we haven't seen any positive plants, wild plants. Percentage of potato psyllids was very low in terms of whether they're carrying or LSO or not, the percentage of positive ones. When you have high numbers, high psyllid populations, for example, in locations such as, example was Alton or Spring Lake, we had a small percentage, but it means that the absolute number of these psyllids that are positive and carrying the pathogen is high. But when you look at the disease incidence this year, it remained pretty low. So the question is why? So we carried one step over, and we looked at the transmission efficiency by single psyllids, and then we also looked at the effect of psyllid numbers. It has been done before, but the difference that the opportunity that we had was also looking at the pathogen quantity in the vector, and also the amount of pathogen they can inoculate. Our observation was that potato psyllids are highly efficient. A single psyllid could have up to 70% success in transmitting the pathogen. We also saw that multiple psyllids are more efficient in transmitting the pathogen than single psyllids and could reach as high as 100%. The interesting pattern was that the probability of a successful transmission event was not affected by the amount of pathogen that these psyllids are carrying. This, to me, indicates that it is a number of feeding events that is important in predicting the transmission success. We also looked at the amount of pathogen that the psyllids inoculated into the plant tissue when they fed. We just removed that plant tissue that they were feeding on, and we looked at the quantity of the pathogen. What we found was that there was no relation. Well, first of all, multiple psyllids inoculated those plants with higher bacterial quantity. But what else? The other thing we found was that the amount of pathogen that was inoculated into the plant was not correlated with the amount of pathogen that these psyllids were carrying, which again indicates that it was a number of feeding events or probing events that was predicting the amount of pathogen that's being inoculated into the plant. We also did this density experiment. Oh, and we also showed that if you have more psyllids on a plant, disease appeared faster than when you had multiple psyllids, than when you had a single psyllid. But when the disease symptoms appeared, the speed of disease progress stayed the same between both single psyllid and multiple psyllid treatments. We also did the density experiment out in the field. We inoculated different cultivars with 5 15 and 30 psyllids. And what we observed that if you had lower psyllid population, the disease severity is going to be lower compared to when you have 30 psyllid populations. And it was just general observation by some of the growers that red cultivars are usually more resistant to disease symptoms. And that was also evident in this experiment that we did. But we are also carrying it over and looking at other varieties as well, just comparing among variety effect to see how they're responding biochemically to the pathogen. And we are hoping to be able to get a better understanding of what are the resistant cultivars or tolerant cultivars, depending on how we look at this or explain it. And hopefully by next year we're going to have a better answer about what cultivars are more susceptible to the pathogen and such vector and what cultivars are not. Dr. Chris Wallace is going to be doing the biochemical analysis, and he's going to be presenting at this meeting. And we're going to continue our collaboration with Dr. Wallace, and hopefully by next year we have a better answer of how this pathogen interacts with different cultivars. Thank you very much.